Welcome to Subculturing Mammal Cells. First, you will put on your protective gear, disposable gloves, and disposable sleeves, and you will spray them with 70% ethanol and pat them dry. Using 70% ethanol and the Chemtech wipe, you're going to wipe down all the material that will go into the fume hood, including your reagents such as your media, PBS, and trypsin. You will also acquire your T75 flask, and you will wipe them down before placing it into the fume hood. Be careful not to touch the filter and never align your filter towards the opening of the fume. You will first remove the T75 flask from the CO2 incubator and observe the overall flask. Take note of the color, clarity of the media, and look for any signs of contamination. Using an inverted microscope, observe the cell morphology and the percent confluence and any contamination that may have occurred. Your flask must be between 80 to 100% confluent in order to subculture your cells. Be careful to not take too long during your observations, as prolonged exposure can kill your cells. As with any material that is placed into the hood, your T75 flask must be wiped down. Be sure to spray your gloves with 70% ethanol before beginning the process. Take out a fresh 10 ml serological pipette and remove the old media. You should tilt back the T75 flask in order to get all the liquid without disturbing the cells. You will then dispose of the old media in the liquid biohazard waste. And you will discard the pipette in the solid biohazard waste. Be sure to spray your gloves with 70% ethanol before continuing to the next step. You will take a fresh 10 ml serological pipette and extract 10 ml of 1X PBS, in which you will put into the T75 flask in order to rinse the cells. Be sure to tilt the flask back before administering the 1X PBS. You will close the cap and swirl the PBS in the flask. Do not get any on the neck or the cap. You will then use a serological pipette to discard the 1X PBS into the liquid biohazard waste container. It is important to note that you do not leave the PBS solution on the cells for too long, or they will absorb it and eventually explode. Be sure to spray your gloves with 70% ethanol if you come in contact with the liquid biohazard waste, or if you remove your hands from the hood. You will now trypsinize the cells. Trypsin is an enzyme that cleaves the adhesive proteins that cells use to attach to the flask surface. You will add 2 ml of trypsin to the flask using a serological pipette. You will then swirl the flask to make sure the entire cell surface is covered. You will then incubate the cells with the trypsin for 5 minutes in a 37 degrees Celsius CO2 incubator. Do not have the cells in the incubator longer than 5 minutes. You will whack the flask to dislodge the cells. Observe the cells quickly so they don't receive too much damage from the trypsin. Using a 10 millimeter serological pipette, add 8 milliliters of fresh media to the 2 milliliters of trypsin cell solution. You will use the pipette to mix gently as to break any potential cell clumps. You should now have 10 milliliters of solution containing cells that can then be subcultured. Generally, flasks are split between 2 to 10 new flasks. In this instance, we will only split 2 new flasks. You will take 5 milliliters of the cell solution and place it into one of the new flasks. You will then add 5 milliliters of new media. That way the flask is up to 10 milliliters of the media and cell solution. Be sure to swirl it around without getting it on the neck or the cap until the entire surface is covered. You will repeat the same process with the second flask, in which you will add the remaining cell solution into the new T75 flask. You will add 5 milliliters of the new media into the T75 flask, and you will swirl it until the bottom is completely covered. You will then observe the new T75 flask underneath the microscope to ensure that there are cells floating around. You will then place it into a CO2 incubator. Be sure to place the lid of the T75 flask away from the opening door as to prevent contamination. You have now successfully subcultured your cells.